Okay, this is video 2, looking at the innate immune system, the second part. And now we are on slide 6 of those slides, looking at circulatory fluids. So our body is full of fluids. There are fluids within our cells that we call intracellular fluid. So much of our cell is just liquid with stuff dissolved in it. We might call that stuff cytosol. We might call all that stuff inside a cell cytoplasm. But there's lots of fluid outside of cells too, extracellular fluid. There's fluid in the blood, and we call that fluid plasma. And you'll see over on slide 7, we have our circulatory system. We have arteries coming from the heart, taking blood at high pressure. The fluid component of blood is called plasma. And that plasma enters these very small blood vessels called capillaries. And the capillaries weave themselves through our tissues. And as the blood passes through the capillaries, lots of that plasma fluid is lost out of the capillaries and into the tissues. When the fluid leaves the blood and enters the tissues, it goes from being plasma in the blood to interstitial fluid in the tissues. Much of that interstitial fluid returns to the blood. Interstitial fluid is very important for how the blood supplies the cells with oxygen and nutrients. So much of that interstitial fluid is returned to the blood, and that's then sent to your veins and back to your heart. But some of the fluid that passes out and through your tissues, some of that interstitial fluid doesn't make it back to the blood vessels. Instead, it is recaptured by the lymphatic system, and in which case it then becomes lymph. So plasma is the fluid in blood, exits the blood vessels out of the capillaries into the tissue, bathes the tissues. Most of it is returned back to the blood, but a significant amount is then siphoned off into the lymphatic system and becomes this fluid called lymph. So onto slide 8, we have our complement system. The complement system is a set of proteins that are found in that extracellular fluid. So they whiz around in our blood, and they also leave the blood into our tissues. They are siphoned off into the lymph, and then eventually all that fluid that makes it into the lymph nodes, into the lymphatic system, is returned to the blood at a different part in the body. So complement proteins, and there's many of them. They are labeled C1 through C9, the C for complement, and many of these proteins have the ability to break into pieces. So the complement protein C3 can split into 2 pieces. It can split into C3A and C3B. Another important complement protein we'll look at is C5, and C5 can split into C5A and C5B. So this complement system has 3 ways of being triggered. We'll look at the 3 pathways of triggering the complement system. And one aspect we'll look at is the production of this molecule called C3 convertase. Now it ends in A's, ASE. That means that we can identify it as an enzyme. And an enzyme is a protein that does some kind of chemical work. C3 convertase does exactly what it says it does. It converts C3 and breaks it into C3A and C3B. So we have this formation of C3 convertase splitting C3 into C3A and B, and that then triggers the system again, and triggers, and we have this kind of snowball effect. We have this activation cascade that leads to the activation of the complement system. And then we'll see that the complement system itself has protective outcomes, and we'll look at those outcomes later. So the first pathway that can trigger the complement system is the alternative pathway. So alternative simply because it wasn't discovered first. The antibody pathway, the classical pathway, was discovered first. So it's not like the alternative pathway is a second choice. It's a very quick and easy to trigger pathway. And we can think of it as kind of an early warning system. Again, if there's bacteria or other microbes in our tissues, we want to get rid of those. And the complement system and the alternative pathway is one way we can very quickly identify and start to destroy invading microbes. So the alternative pathway is triggered by C3b. So C3b is present in the fluids at all times. C3 naturally splits into A and B. And C3b loves to stick to cells. It'll stick to all kinds of cells. It'll stick to invading bacterial cells. It'll also stick to our own cells. But we have regulatory mechanisms that prevent our own cells from triggering the complement system. So you'll see there the picture. What we have is an invading bacterial cell. We have some C3b sticking to that cell, and then C3b stuck to the cell triggers a set of events, making C3 convertase. C3 convertase then splits more C3b into C3a and C3b, and that triggers more C3 convertase, and that triggers more splitting, and that triggers more binding, and that triggers more C3 convertase, and here's that snowball effect as more and more and more C3b gets stuck to the surface of cells. More and more C3 convertase splits more and more C3. So there's the triggering of our first pathway. On to slide 10, we have the second of our pathways. We have the lectin pathway. So one thing that we see on the surface of many microbes is this molecule called mannose, and it ends in O's, so that can identify it as a sugar, or at least it's a carbohydrate. So you'll see in the picture there, we have our purple bacteria. We have our purple bacteria covered in all kinds of molecules, and one of the things that covers not only bacteria, but fungi and other pathogens is this molecule mannose. So we have a protein called a mannose binding lectin. And that's just simply a fancy name for a protein that sticks to mannose. So we have, once again, our invading bacteria. We have our invading bacteria covered in this mannose sugar, and we have a set of molecules that can identify the sugar and stick to it. The binding of mannose binding lectins, or MBLs, then triggers the production of C3 convertase, and that then splits C3, and C3b will start to stick to the outside of our cells, and that'll trigger more convertase, and here's again triggering of that kind of that snowball system, what we call an activation cascade, and then we have a result. And our third mechanism for activation, our third pathway, is the classical pathway. The classical pathway involves these things called antibodies, and we'll spend more time on antibodies when we look at the adaptive immune system. But antibodies are once again proteins, and antibodies float around your body looking for invading pathogens, and then they stick to those invading pathogens. There are these Y-shaped molecules, and the two ends of the Y are good at sticking to pathogens. So we have our invading cell once again. We have our antibodies floating around our body, sticking to these invading cells. These antibodies stick to very specific molecules on the surface of this bacteria, and those molecules are called antigens. We'll talk about antigens and antibodies a lot more later. So these antibodies interact with the complement system, triggering C3 convertase. C3 convertase splits C3. C3b starts to bind to cells, and here's our third mechanism of triggering that cascade, the classical pathway. So on slide 12 there we have a summary, the three methods. The alternate pathway, C3b binds, and then it starts the cascade. The lectin pathway, those mannose binding lectins recognize that mannose sugar on the surface of pathogens. They bind and they trigger the activation cascade. And then our classical pathway, where antibodies bind, they trigger the cascade pathway. And all of those different pathways can all be happening at the same time. You might have antibodies sticking to a bacteria. You might have mannose binding lectin sticking to it. You might have C3b sticking to it. And all of these different pathways can all be triggered. The complement system gets turned on, and then from that triggering event, you get certain protective outcomes. And any one of those triggering events can trigger any one of the outcomes. Okay, that's the end of this second part.